Hi, good afternoon. This is a class that I did several years ago on history of senses. I starting with a general introduction about the history of senses, and then specifically focused on the important and well-established senses such as vision is one section which we'll be doing today. This will be followed by hearing, and then smell and taste are so intricately connected that there will be the last session. Okay, so let's delve into it. Now, history of vision is extremely important in human beings, and as Homo sapiens, we are visual creatures, as we'll talk about when we see the brain representation of your eyesight. And this is why it is so critical that you may recall your first kiss as to why, when you're kissing, uh, people tend to close their eyes because to avoid distraction and any type of, and just because you'd be focusing your eyes or glaring at perhaps another woman or another man who's passing by. So that's one way to concentrate on this, on your partner. Okay, and as I've mentioned many times, that 1/3 of the cortex, the sensory part, is dominated by vision. Okay, so this was, and now eyes are connected with science of optics dating back to Aristotle and Isaac Newton, and even just a refractory error of your eyeglasses. And so it's a very quite involved in science of light, and we receive images at the speed of light. So it's understandably as to how physics is involved. And at the same time, there's we'll talk about some chemistry because after all, the physics uh, is involved with the chemical messages system, and then leading into physiology as to how the signal that is transmitted, with the so-called codes and cones and rods in your eye. And obviously, that's one of the reasons why you may remember your mom told you to take your vitamin A, and so it's a small molecule, but vitamin A is intricately connected with your eyesight and vision. And then obviously we'll get into the more, instead of just basic anatomy, physiology, and we'll get into neuroscience. So the study of senses of vision incorporates many disciplines, as which we'll get into. So ultimately, as you can see from the evolution, that we are all made of atoms and molecules. So we are formed with universe and children of the stars, literally speaking. So, okay, so now the theologians have been involved in terms of how I came about, and that it's still the creationists are still uh, using this as an example, that there has to be a creator because I is so complicated and sophisticated that there is no way it could have evolved. So that's been an argument, and we'll talk a little bit more about it. And even before Darwin, about 150 years before, a theologian named George Berkeley said that it is an opinion prevailing amongst men. Obviously, at that time, the women did count, and all sensible objects have an existence, natural and real, distinct from their perceived being, and only ideas in the mind of. So they were, even as early as that, people were thinking in terms of what are we perceiving, as to how, so that's what the objects exist only as ideas in our mind as to how our brain interprets the images. So they were on the right thoughts at that time. So, and he was later on, he was a contemporary of Locke and Newton. So, and he had written a new theory of vision. Perception is what was most important. Okay. And at the same time, there was another clergyman okay, who was, working at the elite colleges of London, where the common feature of that is a common area which is called quad. So his little ditty that he had written, there was a young man who said, God must find it exceedingly odd to think that tree should continue to be when there is no one about in the quad. So this is again precedes the philosophical axiom or quarry as to if a tree falls in the forest and if there's no one to hear it, does it still make sound? So perhaps we can just say that or extrapolate to if Newton wasn't around, would the apple still be red? Okay, and so similarly, but, I mean that is well understood that obviously the images have no meaning unless there is somebody to receive it and make sense out of it. And at the same time, the philosopher of the Hollywood said that Groucho Marx, who are you going to believe, me or your eye or your own eyes? So that was his, cynical outlook at it. Now, the external eye that all of you know that there is, I don't need to elaborate that, but these are the features, and medically we have different names for it, but they're of academic importance. Superior palpebra, which is basically your eyelid, canthus, and there is a little spot called caruncle, and this is basically now the reason for putting up this picture that not blue eyes are not the norms. It is actually due to mutation in a particular gene, which is a protein coding gene, and that leads to low concentration of melanin, which is the pigment, and so then blue light is shorter wavelength is reflected, and we'll talk about as to, and this may be this like another explanation as to why the sky is blue. It has to do with your wavelength of the spectrum, the rainbow spectrum. Now, interestingly, if we start wondering where the blue eyes are most common, it's not in Sweden or Norway or United Kingdom. 89% of people in Estonia and Finland have blue eyes. Okay, now there are sometimes other various abnormalities that you see. One is called heterochromia, which is the two different color eyes. Okay, and that's considered strangely exotic or attractive. And I think I had a slide somewhere which I don't think I've shared it with this like talk, that Angelina Jolie, the Hollywood actress who was known for the bilateral mastectomy, has different color eyes. Now, more importantly, is the basic anatomy 101 that we need to go over. I think we can all stand a little fraction. Conjunctiva is the outer layer, followed by cornea, which can develop scar tissue. Okay, and then there is something called the anterior chamber and the posterior chamber, which has something called vitreous. It fills the chamber, which is like a jelly-like substance. Iris is, the muscle around it. Okay, and the pupil is the hole. There is a crystalline protein, which is the lens, and the lens is suspended by various suspensory ligaments, which are called ciliary body or ciliary muscle. And the light is now focused on optic nerve, and finally, in the eye human eye has evolved backwards, so to speak. So the part that the image where is processed is actually in the front, not in the back. So it's the design is the outer layer is the tough layer called sclera, and you can just see the outer layer of sclera when you look at the eyes, And then there is the choroid plexus, which are the blood vessels, and the retina is the extension of your brain and nerve fibers. So the sclera is actually hard. Middle layer, there's another middle layer which is called uvea. Okay, and then there are blood vessels that peel through that, Okay, and we already talked about the protein. It is of all this that mesh like retina or retina is, and all these names go back to several thousand years. They were given by the great anatomist Galen. Okay, and iris primarily means rainbow. Okay, and pupils, took until last couple of centuries to know that all of these was extension of the brain. The retinal cells are stimulated by light, and the cones take several years to mature. They are located in the bright spot called fovea, which is the central focus of the retina, and they're responsible for color vision. Okay, and so that's how the image is interpreted, and is then we'll talk about later on of the transmission inside the brain that takes place, which is a complex anatomical arrangement at the base of the brain, and which has a lot more importance in how your vision is damaged in strokes. So we'll get to that in a little while. Okay, moving on. The eye you can move your eyes depending upon which direction you want to look at, because we have front focusing eyes, not side focusing eyes. Again, that has an evolutionary basis as to we are the hunter or the prey. So the rabbit's eyes are different compared to a lion's eyes. And there are varieties of muscles that control the eye movement. And the lateral, which is on the side, and medial, which is on the inside, those are pretty self-explanatory. Superior is on the top, okay, and inferior, that is also top. But interestingly, nature has given us some other facilities called, okay, they're called superior oblique, and so we're trying to understand what the role of that particular muscle is, but there is habit, and these nerves are supplied, are exquisitely controlled by various cranial nerves that arise from the medulla oblongata and the from the brain, from the back of the brain. So, and most of these muscles are now, as medical students, we were to memorize some of these nerves. And the first nerve, if I can, is olfactory, which has to do with the smell. Second is important optic nerve, which interprets the image, which is in the back of the eyes, and it's a sensory nerve. And these are followed by various motor nerves. So oculomotor, again, the word is self-explanatory. It moves the eye muscles, so oculomotor. It controls most of the muscles, except for the two which are outliers. So maybe they have evolved lately. The superior oblique has its own nerve that has evolved, and there has to be some evolutionary. It came in Jonicum lately. Okay, the fifth nerve has nothing to do with eyes, but it's called trigeminal nerve, that supplies your varieties of branches through maxilla and mandible, the big bones, and the sensory. When you touch, that's the trigeminal nerve that takes the impulse to your brain. Okay, and the lateral rectus, lateral muscle, it has its own nerve. Okay, and then the others are related to seventh nerve is facial, and then auditory, and so on and so forth. Okay, but we're not going to give you anatomy lessons here, but just can see how the evolution has controlled the precise eye movement. And obviously it comes in handy when you're chasing an impala on the savannas. Okay, and there are varieties of interesting quotes that one can find in classic literature. Love looks not with his eyes, but with his mind. So very profound statement from Shakespeare. Let every eye negotiate itself and trust no agent. Okay, so again, a very wise statement. Man may see how the word goes with no eyes. Look with thine ears. That was King Lear. Okay, and then finally the statement, which is again very noble for any time. Eye for an eye only makes the word go of blind. So again, so these are some of the codes. So eyes have had roles in philosophy, in relationship with literature for a long time. Okay, let's see, what do we have next? Okay, this is a video which I think it would be interesting from a respectable, reliable source. So hopefully it will play.
Let's examine the eye in detail and see how it works. In principle, the working of the eye is similar to that of the ordinary camera.
When you look at human vision and compare it to insect vision, it's quite difficult to compare them. When you compare them, you really start to appreciate what makes human vision unique.
The lens of the eye forms an image on the sensitive surface of the retina.
The human eye is called a camera eye because the retina is like the film in an old camera. And this is where the lens focuses the light.
Have a look at your eyes. And when I start to look at the back of the eye, I can see the retina.
Magnified many hundred times, the retina is seen to consist of this complicated arrangement of rods and cones.
The rods are really sensitive to light and help us see at night, whereas the cones are adapted to see detail and color. In an insect's eye, there are no rods, cones, or even a retina. Some insects have two large compound eyes which wrap around their head. Dragonflies generally can see almost 360 degrees behind them. Trying to catch them is really difficult because they can see you coming from a mile away. Within the insect groups, dragonflies have the largest eyes, with up to 35,000 individual little ommatidia within each compound eye. That's the thing, they have two eyes and each one's 35,000, so it's almost like 70,000 eyes. It has recently been discovered that they have up to 30 proteins which are associated with their color vision. So when it comes to human vision, we have 3 proteins and we only see red, green, and blue. Our brain integrates that information along with memories. Our brain fills in a lot of the missing pieces. It's only once the raw data has traveled down the optic nerve to the brain that vision is perceived.
So this is the optic nerve, and it takes the messages back to the brain to get processed.
Human eyes are much simpler in general terms. Our brains are much more complicated and can process that visual information to a much higher degree, whereas dragonfly eyes or insect eyes are much more complicated morphologically or externally, but their brains are much more simple.
As you can see, this was a British video. Okay, now a lot of this, the processing was initially the works were worked out by a Swedish ophthalmologist named Alvar Gullstrand, and for that he was recognized with Nobel Prize as far back as 1911. So that's when he combined the optics and the anatomy together. And then again, later on, the variety of problems that eyes develop, astigmatism and accommodation, were worked out. Okay, so this was, and again, this goes down to Euclidean days because everything is geometry. Light travels in straight line, and that helps you understand how the light is interpreted. And this was known in ancient China, and the Greeks had ideas about it. And then later on, the so-called camera obscura became available. And some of these focusing problems were spotted by the astronomer Johannes Kepler, and who correctly hypothesized that there was an element of focusing, refraction took place. That's how the image got interpreted. And then finally, the image is interpreted upside down and is focused with the lens. Okay, and then there people have actually experimented with lenses which are upside down lenses, and then finally retina helps correct, the brain helps to learn to interpret the image correctly. Okay, so and again, this is, so the two-dimensional image that comes out, then we get the depth perception with 2 separate eyes with slightly different focusing and putting it together and making sense out of it. And that's why the eyes are constantly moving. And this was shown by scientist named Alfred Yarbus, okay, and the so-called saccadic eye movements that a physician, a medical student learns to examine this in their first year when they're learning the diagnostic. As you can see, the eye movement of the image. When you're looking at the image, you're not looking at it straight on, but your eyes are moving in different directions. You can just see how your first focus goes on the object's eyes and then on the mouth, and you're concentrating more on different parts, and you have your eyeballs. So the next picture on the right side illustrates how the eye movements take place, and that's called the saccade. Okay, there's like a face perception and recognition, and unless this occurs in split second, okay, and there is a quick movement of eyes. This is one of the fastest movement of your body without your even realizing that you're doing it. As many as 900 times per second. Okay, this is also seen in some other animals, and sometimes there are abnormalities that occur due to balancing problems, and there is a pathological entity called nystagmus that that has to do with the stroke and deciding. Okay, now this is the saccade is a French word, by the way, and that means sail. You know, it's like snapping in the wind.
Saccades are rapid eye movements that can be generated on command, as shown in the video, or involuntarily, as in the fast phase of nystagmus. They are assessed in both horizontal and vertical planes. Ask the patient to hold their head in a central, upright position, keep the head still, and move the eyes as quickly as possible to fix their gaze on an object, for example, the tip of a pointer and your finger. Hold the two objects approximately 20 to 30 degrees apart and command the patient which to look at. Vary the time intervals to avoid habituation. You should observe an immediate, fast eye movement with no delay or inaccuracy on the fixation of the target. Delay, slow movement, disconjugate movement of the two eyes, or inaccuracy in refixation, either undershooting or overshooting, are suggestive of a neurological disorder.
Okay, so that tells you in more details. Now, there are various theories on vision that goes back to the classical Greek era, at least in the Western culture. Plato somehow, for all his genius, got it wrong, that his theory of vision was that the light emanated from the eyes and we throw out a ray which was emanated from the eyes and was, that's how we got, but his illustrious pupil got it right. He proposed a phenomenon called intromission, that eyes actually do receive the rays from outside in. And he proposed that the seat of the vision was in your lens. And then Arabs improved upon the Aristotelian model, the famous Arab physician, scholar, polymath Avicenna noted the importance of the pupil, so they again favored the intromission. Okay, and Descartes was focused on an imaginary object called ether, which was solidified with gaze. Now Kepler finally got it right, that the retina formed an image, and he involved the real physics involved in it. Okay, so there were a lot of other thoughts that went about, you know, and some of them were good attempts, but some of them were hopelessly wrong. Okay, and moving back to the other side, you know, there were varieties of visual allegories that are given by various philosophers and religious people, and so one can say that the dark eye is considered evil, while the light eye is considered enlightened. Yeah, the eyes are open or closed. They have one eye or many eyes. Evil eye versus pure eyes. Goethe, the famous German philosopher, was talked a lot about the optics, and he was. It may be lesser known that he was a man of science, and had proposed varieties of color theories. And eyes are the mirror of the man from inside, and the world from the outside. Okay, and so one can just say that various animals have been associated with different eye features. You know, obviously it's a well-known allegory, as eagles have keen eyes. Healthy eyes are the mirror of the soul. Open eyes are blessed with divine grace. closed eye suggests ignorance and denial. Okay, and then obviously the so-called one-eye cyclops, is well maligned in literature. Again, multiple eyes in the Hinduism, Kali and Shiva has multiple eyes, and there's many eyes on peacock's tail. And then again in images, lowered eyes suggest chastity. Okay, and again, sometimes lust and hate and pride can all been associated with eyes. Okay, at a different stage in literature and a popular culture. Now, the basic question that if you have grandchildren, you may have already answered this or told them to look it up on encyclopedia or Google, depending upon their age. As to this was the question that science struggled with for a while. Why is the sky blue? It is blue because the molecules in the air scatter blue light from the sun. Okay, and that's due to the frequency, or the wavelength rather. Sorry, and so that has a lot to do with how the eyes are reflected in different direction, and that explains how the water molecules reflect light as it falls on it. That light consists of various frequencies, waiting from red frequency to blue, which is the short. Okay, and the charged particles are travel at different lengths. Okay, and this is so that's one of the reasons blue is a favorite stain color of the glasses. And this goes back to lapis lazuli, the precious stone that came from Afghanistan. And similarly, depending upon the sunlight and where you are on Earth, the sky in Finland is more blue than that of equator because of the distance from the sun. Okay, and again, various artists have tried to chase the pure blue color, and they, in fact, so much so that to get a better perspective, they've moved to different parts of the world. And the famous Monet came to England because it gave him a different perspective due to early days of Industrial Revolution. And the same way, painters like Van Gogh and Turner, had totally different perspective, because the colors were totally different in Tahiti versus in dreary old England. And again, these are the two paintings of different way of interpreting blue, as you can see from the cloudy, hazy England. Joseph Turner's vision of clouds is totally different than that of a Finnish painter named Akseli Gavlin. As you can see how the color interpretation of the sky is. So one can say that this is the so-called artistic vision of this, the difference between sight and insight, as to how artists interpret the sky in different ways depending upon their fancy and their location as to where they are. Now, getting back to something that I know a little bit more about. Sorry, one of the pictures may be more gory than you'd care to see, and I should have brought it in later, but not using the slideshow. So apologies for that. The first picture is that of a rather rare disease, but quite serious and can be fatal if untreated. It's called Wilson's disease. That is due to inborn error of metabolism. That has to do with copper metabolism is goofed up. And you can see this a little ring, coppery type of ring that one can see. So if you see, you can look like a very good diagnostician if you make a diagnosis of Wilson's disease just by looking in the eyes. And now recently, if you have seen late night television ads, that due to thyroid disease, where there is too much thyroxine being produced by a tumor in your thyroid gland, your eyes start bulging like the former Hollywood actor Marty Feldman, the bulging eyes called exophthalmos. So the eyes are bulging, and that's the diagnosis that one can make of overactive thyroid, along with other signs that take place. Now, this is unfortunate, is very rare, that sometimes tiny worms can, I think it is skinny worm, if I'm not mistaken, that can travel along the bloodstream and can get all the way, end up in the eyes. and it's a pretty gruesome way of, fortunately, parasitic disease is far less rare, but the treatment can be very difficult. Okay, and then again, every clinician would learn to look at your eyes. Nowadays, obviously, people do not bother examining patients, but if you're a good clinician, you can look at the conjunctiva and get a rough estimate as to how anemic a person is. And these are the varieties of light levels. Even a brown-skinned person can have a conjunctiva, can look awfully pale if your hemoglobin is markedly low at 7.5 grams. The normal being 14 to 16. On the other hand, if you have a nice pink conjunctiva, then you can see that would be a nice, a red eye that you can see in the last picture D, sorry. And the other in between layers, the B is just slightly different, so one does not need to be a genius to be able to tell that as to how it should be, so you can just, a rough estimate that one can avoid, it can be ahead of your labs. Now, initially, science has a lot of false starts that goes through, starting from bleeding George Washington to cure his quinsy, that is the abscess on his tonsils, to examining your bumps on your skull and trying to see what type of personality you have or whether you. So for a while, there was some enthusiasm about iridology. So iris were considered windows on the soul, and by various iris pattern reflected patient's health, and people went all out and created varieties of fancy charts of so-called iridology, the zones. So this was popular, in the past, and you can see 1665, Chirometrica America. So you can see how this was popular for a while. And then again, then it again caught on in the 19th century, and by a Hungarian physician, some streaks in the eyes, what they saw as to. So this was eye disease diagnosis was, you can see how they were trying to make sense out of this little, blood marks. Okay, and this was before the invention of, and there was also a craze for trying to diagnose, called urinoscopy. You're looking at urines and trying to diagnose. So people have, for God knows, they have tried to make some sense out of the complexities of human body. So varieties of crypts and folds, so, but there is value that the iris is very individualized and can be used for identification. And those of you who are James Bond fans may remember that from one of the movies, and I think it was Thunderball, where they were impersonating pilot, Air Force officer, and they take picture of the fake eye and the eye recognition. So in even your camera now, it can base your face and your eye recognition. Identification can be established, and this has been well used in various movies, starting not just from Thunderball, but Blade Runner and Minority Report. Okay. So Hollywood has made, okay, and now this gets down to the Darwinian right that I alluded to earlier, that theologian, named William Paley, okay, implied that there has to be a watchmaker because the eye is very complicated and could not have evolved, okay, on its own, in spite, with all due respect to Darwin. So this was an argument that was proposed and encountered for argument for design in Patients of Faith. And now Richard Dawkins, the famous atheist and geneticist, has systematically torn down the arguments and had a book which was popular back, I think, in the late 80s or early 90s, called The Blind Watchmaker. Okay, and so there is a nice review of that. Okay, so this now, and as soon as Darwin had written his book, he was questioned by a physician who was a believer in Darwin's theory, but he again, had written letters to Darwin regarding the origin of the eyes. So Darwin was bothered by the legitimate concern that Asha Gray proposed as to this, and his reply was, when I think of it, tells me that I ought to conquer this cold shoulder. Okay, so the eye with all its inimitable contrivances for adjusting the focus at different length, different light, amount of light, correction from spherical, and so they were far ahead in their optics by this time. When okay, so I feel confess absurd to the highest degree. So he was again flummoxed with how this because he did not have the advantage of comparative genomics and the molecular basis that we have access to. So and now this interestingly, those of you who know a little bit of genetics are well aware that on the other side of Europe, Gregory Mendel was proposing genes, and that might have given him some ammunition as to how the evolution works at a microscopic and genetic levels. But again, those was days before internet, so so Darwin was flummoxed by it, and this is again has been. I talked about it as to how on one side we have Darwin, who talked about the endless forms most beautiful. This is not his book, by the way. This is a book by a biologist at University of Wisconsin. He talked about evolutionary parallels, starting from eukaryotes, the basic cells, to humans. As to how the evolution took place and was not a gradual process. And he goes into details about it. So it was an interesting read for evolutionary tree that we have learned. And at the same time, this was the good work by Richard Dawkins, who again supports the genetic or genomic explanation for human evolution, dating back to the earliest origin of life on planet Earth. So this can all generally be explained with natural selection, and given enough time, the complex adaptation can occur, because nature, as some of you with a background of science and genetics would know, that nature is very conservative, and a gene that is that's why the geneticist can study fruit fly and can make sense out of what type of genetic code has been preserved all through the evolutionary tree. Okay, so so Dawkins' first concern was the difference between potential of complexity and pure randomness versus cumulative selection that takes place. So okay, so and this has all been now well worked out, after another 40 or 50 years. Okay, now again, the examination of eye was difficult because when you examine the eye, you can't shine light in your eye because your own head comes in the way. Okay, so later on, right, with the advance of optics and lens technology, two physicists, obviously Charles Babbage was not just a physicist and ophthalmologist, but was also the pioneer of early days of computing before electricity, the Babbage counting machine. But we are digressing. So he showed it had created an instrument for looking at the inside of the eye. So basically, mirror with silver and fixed, or how you can just see the tube at an angle. So this was the ophthalmoscope for looking in the eye, invented by Charles Babbage. On the other side, the Germans were almost parallel as to how it has happened quite often in science that two inventions have occurred more or less at the same time. I mean, the classic example would be Edison and the British scientist who discovered light bulb more or less at the same time. Sorry, I forget his name. And so a lot of other discoveries have occurred more or less by sheer coincidence. The classic example is Darwin and Wallace, who both came up with the idea of evolution, 5,000 miles apart. Okay. Darwin was first before, Wallace, but again, one was not aware of other person's work. So Helmholtz was a little bit more sophisticated system, perhaps due to better lens technology in Germany, and eye mirror, looking through the eye, through the back of the eye for reflection, and the clear image was focused with a combination of mirror and lenses. So this was an ophthalmoscope, which is a standard an instrument that a lot of medical students learn how to use. Okay, so a retina was difficult, as we talked about, to examine, but now Helmholtz had invented it, and again, it said, well, what would it be used for? oh, this is a splendid toy. So he didn't put two and two together, but later on the invention caught on very quickly and the medical establishment started using it. Okay, and this is just the basic normal picture that one would see. The fovea, which is the central part of the retina that is responsible for sharp central vision. And the fovea, just the Latin word, simply translate as a pit. It contains about 150,000 cone cells. So there is a large concentration that leads to the sharpness of the picture. Macula is a red-shaped area, oval-shaped area surrounding it, about half a centimeter. Okay, and it's a central high resolution responsible for color vision. And the macula lutea means a yellow spot surrounding it. Macula absorbed an excess amount of blue and ultraviolet light. Okay, and it contains varieties of pigment. And if there is damage to the macula, then the central vision is lost. It can be due to trauma or due to loss of blood vessels. Okay, and this is the optic disc inside. Now, well, the nerves and the retinal blood vessels are there. And so this is just a normal picture of the beyond iris. Okay, and now let's talk about another thing called blind spot, which is not there is actually a genuine entity, not a blind spot, as to in common literature.
Full screen. Close or cover your left eye. Look at the plus sign. Be aware of the circle, but don't focus on it. Keep looking at the plus. You may need to move your head back and forth a bit or move your thing closer to your face, but at some point, the circle is going to disappear. Now close your right eye and look at the circle. Move your head back and forth until the plus sign disappears. You have just found your naturally occurring blind spot in each eye, and of course, in daily practice, we do not notice this. The human eye has what you might call a kind of a fundamental flaw. Light-sensing cells in your retina send signals to your brain via nerves, and those nerves are in front of the light-sensing cells. Eventually, those nerves have to pass through the back of your eye to get to your brain, and in the part of your retina where they pass through, there aren't any light-sensing cells. That's your blind spot. Now, this isn't normally a problem because the blind spots are located at slightly different points in each eye, and each of your eyes work together to fill in a complete picture. But even with one eye closed, you're not seeing a big black hole. Instead, your brain fills in what it figures ought to be there. That's why when the circle disappears, you see the color of the background. Your brain is guessing, and it's guessing wrong. Although at least one very small study found that you might be able to shrink your blind spot with practice. Researchers showed 10 participants an image that fell within the margins of their blind spots and asked them to describe it. By the end of the experiment, people got a little better at describing those images. The researchers think it's because the light-sensing cells right around the edges of the blind spots became more sensitive, better at picking up and passing on light signals. That's the kind of skill that's probably not gonna ever make any kind of difference in a life or death situation. And humans have had blind spots in their eyes for as long as we've had eyes. But it's a neat way to try and hone your brain if you're into that sort of thing. There is a different kind of creature, though, that just completely avoids this problem. Cephalopods, like octopuses and squids, have their nerves behind their light-sensing cells, so there's no need for them to have a blind spot. Why did we not do it that way? Evolution. Well, I, for one, welcome our tentacled over.
Okay, so that was an interesting way that, again, now these are some of the pathological images that take place when the retinal artery, there's occluded. It's a tiny little branch, but again, when it causes, due to stroke or cholesterol plaque, you get a lot loss of vision, and there are various shadows that you can just see on ophthalmoscopic examination. Now, sometimes, that nature does the opposite trick. There are new vessels that form, primarily in diabetes due to too much glucose and abnormal metabolism at the local level. And again, the varieties of other illnesses such as sickle cell disease can cause problems, and macular degeneration just with a simple aging process becomes a thing. So these are the varieties. You can just see that you don't need a medical degree. You can see these are the areas, new blood vessels called angiography, leaking vessels. And so this is the various abnormalities can be diagnosed because we are getting a direct view. Okay, now let's I think we have something about going back to the earliest primitive cells.
Have a red-colored feature called an eye spot. At first, it was thought that this was a light sensor until extensive observations reveal that this red eye spot was actually blocking light in order to establish the direction of light entering the real light sensor. Recent studies have clarified the ultraviolet activation mechanism of the light sensor on a protein level, an important discovery expected to assist with the clarification of neurological functions. That doesn't mean all mysteries, however, have been solved. Under a microscope, euglena can be observed avoiding dark regions rather than gathering in patches of light. This points to the existence of a sensor for measuring the intensity of visible light, in addition to the already identified ultraviolet sensor.
Okay, so that gives you an idea about the evolution even in primitive organisms. So the photoreceptors and pigment, which provides the energy to get around and sense, and reach for food. Okay, so with the bright light, it creates photosynthesis. There are varieties of protein which you may have or may not have heard about, called rhodopsin and flavin. So these are the lens protein that are conserved as far back as in bacteria, and this pigment is same as even in butterfly wings, and that makes the cat's eye grow. So again, similarly in fish swim bladders. So eyes have been around and evolved for almost half a billion years. So some of the proteins have evolved in different length and different complexity in different animals. Okay, and as we saw in this slide, videotape, we saw the euglena as light detection, but not real vision, so to speak. So, and again, these are very tiny, early, organisms. So the eyes have, been around for a long time. Now there are varieties of body plans, for formation of eyes. So the image-forming eyes, basically. So there is no one design across the animal kingdom. So there are less than different eyes. The insect eyes, hundreds of miniature eyes that we already talked about in Drosophila, eyes of squid and octopi are different, but there are vertebrates. They appeared independently, and we talked about the changes that are seen. Okay, so and the light sensing molecules developed at a different stage of life. They got folded into varieties of membrane and for capturing photons as they enter. When they're in a font, that these cells starts responding, okay, to different directions, and then the pigments evolved, and then finally the nerves that send information to the brain. Okay, so varieties of pigments. We talked about rhodopsin. Similarly, there is chlorophyll. There are varieties of proteins have developed. There are some animals have even magnetic field detection. Okay, so this is various type of convergent evolution. There have been 10 different types of eyes that have evolved with different molecules, and that eventually this light is converted into electricity, and the message is transmitted through nerve endings to your brain to make sense out of it. Okay, all right. And a lot of these evolutions occurred during the so-called Cambrian explosion phase. Okay, there were predators and prey. I showed how the position of the eyes can be different depending upon direction of the light and the resolution that takes place. Okay, and finally, the more complex eyes evolved in making sense out of what they are seeing, such as facial recognition, and able to, get a more precise resolution, and that helped in pairing and reproduction, obviously. The earlier forms were primarily oriented to landmarks and object avoidance. So you don't want to run into something else, So that as this evolution went from a different direction. Okay, and all of these was controlled by a so-called master control gene called POX8. Okay, and this mutation, if there is abnormality in that, it leads to absence of the eyes called aniridia. Okay, and this is the so-called master control that develops, sensory organs. It's similar in starting from the lowly zebrafish to diverged, from human development. The change occurred around 400 million years ago, but the gene itself is it's conserved across varieties of species. So that's and instructs the cell and to different types of embryonic development that takes place. And okay, and see if we can see that the wild type, the WT is called wild type, and if there is mutation in this thing, you can just see that clearly the eye looks abnormal. There's the lens is abnormal. Similarly, in zebrafish, also the eye is decreased in size and lenses decrease, and the retina is malformed. Same in mice and in Drosophila, which is the so-called eyeless fly. Okay, so the scientists have a privilege of naming themselves. Okay, and so this so one gene has a huge, long-standing reach, the eye formation. Okay, and and we already talked about how vision is an important part of the brain. So in this particular gene has a dramatic role, and it's also not just involved in the formation of the eye, but has a role to play in frontal lobe development of your brain as well. Okay, so that's so much for genetics type of thing. So now again, this is an interesting thing. We'll talk more about it when we talk about animal senses, but I think I'll just show this to you as long as I have it.
They really are beautiful animals, really smart animals. Very large brains for body size.
It's hard to hide in the ocean, but the Hawaiian bobtail squid is a master of disguise.
During the day, if they're disturbed from the sand, they'll come out and just sit on the surface with a sand coat on them, trying to be invisible. And if that doesn't work, they'll eject out ink and leave that blob of ink behind in their shape.
Their camouflage strategy at night is even more amazing. Luminous bacteria actually live inside the squid's body and light up its underside, helping it hide from predators.
So they're emitting ventral light. They're camouflaged against the moon or the starlight so that they don't cast a shadow. And so as not casting a shadow, it's like a Klingon cloaking device.
With support from the National Science Foundation, biologist Margaret McFall-Nye studies the squid's remarkable light organ, the small pouch that's home to the cloaking bacteria Vibrio fischeri. Its structure has a lot of similarities to an eye.
We can look at all parts of the light organ, so all parts of where the bacteria go into the light organ and then reside.
To better understand how the bacteria take up residence inside the squid, McFall-Nye's team studies the babies under the microscope.
These protrusions right here, which we call appendages, help to filter, basically bring in bacteria from the seawater. So, you kind of have this little cave, kind of nest that the squid makes for the bacteria.
We bring the eggs into the egg room.
Nell Bakaris goes to Hawaii 3 or 4 times a year to collect adult squid off Oahu. Back in the lab, she oversees the squid condos and the nursery.
I mate the squid every 2 weeks. A female will generally lay eggs about every 3 to 5 days. This clutch is a fairly good size, probably 200 eggs or so.
Analyzing this squid bacteria symbiosis could help develop new camouflage materials for the military or lead to new pharmaceuticals to fight bacterial infections.
At the very basic level, we're asking how animal cells talk to bacterial cells.
A conversation worth listening in on. For Science Nation, I'm Miles O'Brien.
Okay, and so this has to do with the number of bacteria that are present. That's called quorum sensing. So that's an important concept in bacteriology, by the way. Okay, and this has, again, goes down to the gene called luciferase gene. It's a gram-negative gene. Okay, and so this is, the camouflage gene. Then there are some other varieties that this toad was born with eyes in the roof of its mouth. So there are varieties of macromutation that takes place. Okay, I don't think this is not a video. I think this is just an image, by the way. So it's a curiosity type thing. So, and so this is like a improbable amount. So how genes and evolution works in a mysterious or different ways. Okay, and we talked about the eyeless gene, so the eyes can be in different position depending upon the mutation that takes place. It can be on the legs sometimes, and a lot of people who are doing the so-called bioelectricity experiments have a way of growing eyes on different parts of the body. Similarly, the whole leg can grow in different part. And so the body plan can be altered with morphogenesis. And if I ever get around to doing a class on bioelectricity, we may talk at length about it. And so some of these researches were well recognized by a Nobel Prize for the so-called embryonic induction that takes place of the body plan formation. And so the varieties of gene controlling proteins called transcription factors have which they bind to varieties of genes and cause this expression of formation of the so-called body plan as to between right and left, front and back, north and south type of thing. So, okay, and so these are they are much more universal type of control, but the simple variation or changes can have a devastating outcomes. Okay, and again, so this eye evolution, so varieties of computer scientists have become involved in studying.
I've been interested in the question of how long time it would take for an eye to evolve.
Nilsson envisioned a sequence of stages by which a flat patch of light-sensitive cells on an animal's skin could evolve into a camera-type eye. As a first step, nature would favor any changes that made the flat patch more cup-like.
As soon as you created even the slightest depression in the center, it means that the edges of the cup will actually shade light from parts of the environment. And of course, all the light-sensitive cells in this little cup, they won't measure light in exactly the same direction. So already this cup has some pictorial information.
Another model demonstrates what a primitive cup eye can do. The brightly lighted skulls cast an image onto a translucent screen Nilsson installs at the back of the cup to act like a retina. But the image is not at all well defined. The cup eye can do little more than detect movement. This kind of eye can be found in nature today, in flatworms. Their eyes evolved no further. In their environment, that's all they needed.
But if the animals need to move faster or evolve to become fast predators or to see other fast predators, then the construction needs to be improved. And one way of doing that is to constrict the opening, to make it smaller.
That's just what happened to creatures like the chambered nautilus. Over thousands of generations, natural selection favored those with slightly more constricted eye openings, which focused light more sharply. This worked well, up to a point.
Since this strategy of making a sharp image also has the drawback of creating a very dim image, it's not very popular in the animal kingdom. And there is an alternative solution, which has become very popular in the animal kingdom, the solution that we use in our own eyes, and that is to put in a lens.
Nilsson's model lens uses two thin layers of clear plastic. He can inject water in between them to make the plastic windows bulge out like a convex lens. This mimics what natural selection might have done over a few 100,000 generations, favoring animals with a rounded, transparent layer in their eyes that cause light to be focused more.
Okay, so I think it's a long, this scientist has almost a 1 hour worth of talk, I think there's a link here later on, but it's for sake of time, we're not going to dwell too much onto it. Now, the eyes are very adaptable as to see how the dimple eyes, the pimple eyes, okay, and for gathering eyes. And the largest eye we'll talk about more in the next class that formally starts in October about the praying mantis. These had some of the biggest eyes with the eyeballs. And so it's an interesting animal that we'll talk about at length, but it's a mean little creature, okay. What you doing? Yes, recording. Okay, and here we already talked about the compound eyes of the dragonfly, so I won't talk too much about it. And various insects and crustaceans have that, so they're a poor resolution, but they're better at detecting motion. So, okay, that's the point that I think we don't need to elaborate it too much on it. Okay, and this is again another nice example of the dragonfly eyes that we already talked about at length. Sharp vision due to large lenses, etc. And humans have, now we have trichromatic vision, red, blue, and green. And there are some people who are so-called tetrachromes, and I'll have some example of it later on. Okay, and there are varieties of proteins that we talked about. Okay, and these are the scallop eyes. So let's just show this. I think I'll again show it more, but I will just show this. There's no talk on it, but you can see the scallops as multiple, two sets of reflecting eyes, and so one for floating things by, another for high definition for running fast. Okay, so this is, so another, miracle of evolution that you see. Okay, and the largest eye are that of colossal squid, which are about 10 inches wide. Okay, and some of the other animals have changing color with different seasons. For example, in reindeer, okay, some fish have multiple 4 sets of eyes. And camels have 3 eyelids. By the way, the praying mantis has overdone it with 16 types of color receptors. Yes, oh yeah, and here this is the giant squid eye. You can see it's almost as big as a coconut type thing. So, but the squid itself is a giant of the ocean. It's about weighing 1 ton. So, and the optic center is not that large, type thing. But so these are huge number of examples of that I can go on for at length type of things. But I think I'm just going to enclose it in a slide. So I'll read only some of them. Dolphins sleep with only one eye open. Dragonfly we talked about, and geckos can see colors around 100 times better than humans. Okay, and chameleon have independent eyes from each other. Okay, and camels have different 3 eyelids to cover for sand, basically. Okay, a dog's vision is different. They can only see in 2 colors. Okay, and cat's eye have a almost 285-degree sight in different 3 dimensions. For peripheral vision for hunting. Okay, and so this is that's a huge variety across the whole animal kingdom. Okay, and these are some of the other examples that we already showed you, that thing. So, and the so-called convergent evolution that takes place. Now, this is the one slide that I cannot show because of copyright. And some of the diving culture that's in Southeast Asia have developed, have evolved their eyes in in a short span of time to see clarity underwater. So that's a BBC video, which I cannot show. Okay. All right, and xerophthalmia. Now this is already, you already know about the lack of vitamin A causes dry eye and blindness. So diet was suspected, and this was discovered in the 19th century in the relationship with, and this was again very common during World War I, and that led to the widespread use of cod liver oil. Some of this mechanism as to how this was obvious based on observation alone. Okay. But the actual chemistry and the physiology behind it was discovered by a scientist named George Ward as late as 1967. The varieties of protein, retinal, and vitamin A relationship, as to how this was protein changed shape from purple to yellow to dark blue to purple again. Okay, and so an important role of vitamin A was he would establish, very clearly, and that was recognized with Nobel Prizes. And again, the philosophers and the mystics have made a lot of things about the awareness of opening the so-called third eye. That some of the work by Carlos Castaneda was very popular, and so this is, and we already talked about Julius Axelrod's Nobel Prize. And the importance of pineal gland and the melatonin pigment, which has to do with your body clock, internal body clock. that was again very, we talked about it when we did a class on science of sleep, okay, the importance of melatonin and establish the day-to-day. And this again explains the layer of the eye in a more schematic diagram. As the light comes into the eye and it goes, this is the sclerize from outside in, and they exposed to the retinal pigment, and finally the in between cells, have no way of light, and then the axons that are in the front, and they're transmitted to the optic nerves, as we already talked about how the eye, and there are the cones and rods that the rods and cones are laid out in nicely, and they are combined with something called the horizontal cell. They're the bipolar cell, which have established connection on both ends, and there are other types of cells which have, they're all this. I mean, right now, I'm sorry, I didn't make notes on it. And then it transmits the impulse to the ganglion cells that take message to the optic nerve, which is in the center of the eye. So, and how the human eye is backwards, it's extension of the brain. Okay, as it took place, and and so the rods and cones, the numbers are different. The only cones are relatively fewer in number, only 7 million. Okay, while the rods are black and white, and they're in huge number, 125 million. And so there are varieties of cones, the dark red and pale green. Okay, and so depending upon the time of the day, the color cells will respond differently, type thing. So, and then again, rods are blind to red eye. That's why the instrument panel of the airplane is of that color. Okay, and so there are cones are kind of like a hotline to the brain, I think so, and they're connected to bipolar cells, as I already alluded to. Okay, and and the first person to draw this beautiful drawing based on the anatomy, microscopic anatomy, was the great neuroscientist Ramon Y Cajal. Okay, and he was an artist, and not only just a great neuroscientist, but also an artist par excellence, and and a neurologist. Any neurology talk will have a nod to his drawings, which were exquisite. Okay, all right. So, and now the retinal ganglia respond differently. Okay, to the on-center, off-center response. So I think I have a slide for that, with depending upon the stimulus, on-center of retinal ganglion cells and off-center. So depending upon the dark objects are as common as light ones. So depending upon that, the people who discovered that moving away from on-center, the spontaneous rate is inhibited, because both these types of cells are equally distributed in the eye. Okay, I think I have a video centers around sound picture.
Light hyperpolarizes photoreceptors. The signal propagates through bipolar cells to retinal ganglion cells, or RGCs, which carry visual information from the retina to the brain. RGCs have a receptive field that can be described as two concentric circles, a small circular center, and a broader ring around the center called the surround. These receptive fields fall into two categories, the off-center on-surround and the on-center off-surround. In RGCs with an on-center off-surround receptive field, when there is no light stimulus, the RGC fires at a baseline rate. When a light spot coincides with the on-center, the firing rate is at a maximum during the stimulus. As the light circle expands to cover the off-surround, the firing pattern diminishes closer to baseline firing. When there is light only on the off-surround and not on the on-center, then the baseline firing is suppressed during the stimulus period. The receptive fields of RGCs are established by signaling in upstream retinal circuits. There are two types of bipolar cells that signal to RGCs, on and off. Off bipolar cells preserve the sign of the cone and are therefore hyperpolarized by light. On bipolar cells reverse the sign of the cone and are depolarized by light. In addition to sending signals to the bipolar cells, cones also send signals to horizontal cells, which connect to surrounding cones. Horizontal cells receive excitatory input from the cones and send inhibitory feedback in return. In this schematized on-center off-surround circuit, a center cone is flanked by surround cones and connected to an on bipolar cell downstream. When only the on center is illuminated by light, the center cone is hyperpolarized and therefore releases very little neurotransmitter. However, the surround cones are depolarized because they didn't receive light and therefore still release neurotransmitter. The horizontal cell is excited by this neurotransmitter and continues to send high levels of inhibitory feedback to all the cones. This inhibitory feedback causes the center cone to release even less neurotransmitter. The on bipolar cell, postsynaptic to the center cone, is maximally excited as it reverses the sign of the cone. When the light expands into the inhibitory surround, the surrounding cones become hyperpolarized, and the horizontal cell is less excited. Inhibition to the center cone is reduced, and the on bipolar cell is consequently less excited compared to the previous state. When light reaches only the off surround and not the on center, the surround cones release little neurotransmitter. Since they are more numerous than the center cone, the horizontal cell is minimally excited, and its inhibitory signals are low. The center cone...
Okay, so you get the idea. That's a very complex interplay of different layers of cells and stimulus on and off and the workhorse of. So you can see that is a role of electricity, which is what she means by depolarization, polarization, and chemistry, which is the neurotransmitter, which, you know, the workhorse being acetylcholine. Okay, so this way your eye is looking for contrast, and okay, that is formed, and that's one of the reasons how we can distinguish the images. So, and that's why, and so motion is detected first in black and white, then the shape and form and depth. So that's why the two eyes work in close collaboration without you realizing. Okay, now the important impulse is transmitted not from the optic nerve directly to the right side, left to the left side. It's a lot more complicated than that. And again, the famous anatomist Galen devised the terminology called chiasma, which the optic nerves come together, okay, and they remain on their side, and then finally they switch slides. So that's the optic. We'll talk a little bit more about it, okay, and so there is. Then Descartes also proposed that the lenses reverse the image falling on the retina. So there is a partial crossing that was emphasized. So basically, the change that was taking place in vision, as the brain map was coming to the picture. Okay, and this led to the English neuroscientist, okay, to work some of this out. The neurophysiology of the optics was worked out. As you can see, let's start with the left visual field, the temporal side, which is which is the outside nasal aspect, and you can see that the impulse is formed in the left eye, okay, and in the temporal part, okay, and the fibers stay on the same side here, okay. You can just see that, the image that forms, that stays on the same side, the dark green, and the light green, okay, crosses over to the other side, and that's called the optic chiasma. So the medial aspect, the fibers cross over, okay. Each visual field is divided in two parts. Passing through the lens is projected on the opposite side of the retina, okay. And in the optic chiasma, again, the fibers cross over, okay. And so this is, the optic tract, and this is the optic nerve here and the optic tract, and this is how the image is projected on the opposite side of the brain, okay. And this has a lot to do with where the stroke occurs, that depending upon where the damage is, as to which side of the visual field will be paralyzed. For example, the middle cerebral artery that causes damage to the optic chiasma, so it will cause called homonymous homeanopia, means that the outside of one side is damaged, where the inner side, the medial aspect, is damaged on one side. So that's the classic stroke that takes place due to something called the middle cerebral artery. Okay, and this was worked out by the circle of blood vessels by the famous English neuroscientist Dr. Willis, called the circle of Willis. Since this is not a brain lesson, I didn't put that on top, but the circle is surrounding, depending upon the anterior, the medial cerebral artery and the posterior cerebral artery. They supply different parts of the brain, and depending upon that, we can diagnose the anatomy as with the clinical findings that can be diagnosed at bedside. So the optic nerves are connected to each other at the chiasma, which is the crossing that takes place from one side to the other. The temporal side stays on the same side, and beyond that it's called optic tract. And these fibers project in a varieties of nuclei, which is a cluster of neurons on one side of the brain. It is called the lateral genicular nucleus of the thalamus. Thalamus is like the relay center of the brain, and from that the image is projected into the visual cortex, the so-called optic area, and then again to the front of the brain to interpret the image. So the visual perception theories have been around for a long time. This is the brain, which looks like the big mass. And again, the various anatomists had a different course, how we drew type of the picture type that depends upon. So these are varieties of this is English cartoon thumbskulls, which I think is not too familiar with Americans. Okay, and but the basic principle that was worked out, that our perception does not identify the outside world as it really is. We're allowed as a consequence of transformation performed by our senses. So the photons are converted into images, vibrations are converted into sounds, a chemical reaction to smell. So this is how the universe is actually silent, but we make sense out of it based on varieties of mechanisms by which the image is interpreted. Okay. And this all became available within the last 100 years. So there is no little guy who is trying to analyze the image. So-called eye department, nose department, ear department. There's no individual, but basically these are varieties of stimuli. That's one of the reasons for this class. And again, one can see how the retinal image that comes into the brainstem area, and it's transmitted to the it's like the so-called optic. This is the occipital or the back part of the brain, and then the other big transmission occurs, goes to the higher visual centers up in the cortex, and in the sensory cortex, which is in the middle, which is so-called where, so that perceives the depth, the figures, and so initially we recognize the image, and then we try to make sense out of it, and so there are two main streams of nerve fibers that take place from the eye. So to where and what pathways that take place. And a huge area of cortex is devoted to it, as I talked about earlier type thing. So, and the massive area is devoted to central vision. Okay, in the cortex, of varieties of specific cells. And for a while in neuroscience, when they were doing epilepsy research, they were able to stimulate the area, and the person who was obviously brain does not have any pain fibers, so the patient in that case was awake. So it generated the image of the popular actress at that time called Jennifer Aniston. And so one can say that in brain there is so-called Jennifer Aniston, and some people got stimulated the image of President Clinton, the same way there is so-called grandma neuron that is there. So varieties of areas. So the image interpretation, the brain's learned behavior is very specific. It can be localized at the time of surgery. So these are the pathways were confirmed. The war becomes a big area of research, and these are the varieties of part. The brain cortex that are the posterior part and the lateral genicular nucleus that we talked about, which is at the junction of the temporal and the occipital area. The ventricles are the empty spaces of the brain, by the way. Okay, and okay, so this is more neuroanatomy than you'd care to know. So the brain does simultaneously allows you to interpret color, form, depth, and motion. Okay, and we talked about the blind spot that we already talked about. Okay, and a lot of the physics that we talked about was the respect to the classic experiment of Newton, and who proposed the light as a particle. And then at the same time, the theory a few years later was published by Young, while he proposed the light as a wave. And theoretically, it's probably they were both correct in their own way, and that the wave theory was favored by the European school of physicists, and the particle was the Newtonian. So, and the wave interference was created, and this is the classic burst down to the quantum physics. And this is the well-known, as you can see, how our length is quite limited to visual spectrum, occupies the real narrow spectrum of light, that and the narrower wavelength starts from the gamma rays, which are dangerous and can be harmful to your body, followed by X-rays, the ultraviolet light, and the so-called infrared. And the visible spectrum is, ranged from only 380 to 750 nanometers. And the longest frequency are radio waves, Okay, and this was all well worked out, the classical spectrum, the red, orange, yellow, green, blue, and violet. And later on was indigo was added. Okay, and the varieties of and Thomas Young measured the wavelength. Okay. Now, interestingly, some bees and insects are able to see ultraviolet light, and so can some birds. While on the other hand, snakes can sense infrared light, and pythons had to evolve to catch prey because they are cold-blooded and based on thermoregulations, so that their evolutionary pathway is different. Okay, and again, when it comes down to how the brain perceives the image, how the image is processed in different parts, and perception. The PET scanning, shows the imagery, in processed in different parts of the brain. Perception is driven by imagery is based on what has been perceived before. The perception is driven by direct sensory input, and so this is what the artist, rendered. And now moving on to color vision, which is quite complex way of processing because the image is broken down into lines and shape. First, way the various layers of the brain. There are like 6 layers for color, and so the scale it's almost like in a company hierarchy. The images evolved, and lines and shapes are processed first, followed by motion, and then later on color and form are perceived. Okay, so there is some delay involved in interpretation of some of these images, and we already talked about, and finally the image is upgraded to the 4th and 5th layer of the brain. Okay, and so finally the image is projected, so we can just see how the basic image of lines and shape, and that is elevated to color interpretation. So color interpretation is, it's quite a complex process, and so this is another way of
so green. I see them for me and you, and I think to myself, what a wonderful I see skies. Y'all say good night, and I think to myself, what a wonderful world.
Okay, so you can see how the great Satchmo said, the different frequencies are interpreted in different ways, the blues and the reds. Okay, and again, this has a lot of implication in the theories of consciousness as to how our brain interprets the color as red, as to impression, and there are various philosophical writings have been done as to people who have totally blind, has never seen but knows everything about it based on theory as to what their perception would be. So it has a huge implication as to make sense out of the evolution of consciousness. Again, then we talked about the infrared and ultraviolet light that dates back to William Herschel, who did varieties of experiments of sunlight, and again, made discoveries of infrared and ultraviolet spectrum of light. Now, getting back to the medical aspect, there are some of the commonest abnormalities: the myopia, the presbyopia that occurs, and hyperopia. Okay, so the short-sighted, long-sighted, as to the shape of the lens changes, either just due to a defect in the lens, okay, and the thickening, inability to change the shape of the lens, that and presbyopia that occurs with aging process that we're all familiar with. Hyperopia is the so-called farsightedness, okay, which makes objects blurry and cause headaches, eye strain, etc. Myopia is, classical way of nearsightedness, and as you may be very well aware that the epidemic of myopia is increasing, especially in Asian countries, that almost 90% of the Korean children have myopia and require glasses, and that may have to do with the emphasis on learning and being in school. So it is encouraged to reduce that, that from time to time you should take a break and look at the outside. Okay. And presbyopia, again, makes it difficult to see things up close as the image is focused behind the retina. Okay, and so you don't have extremely long arms, so that occurs due to lens hardening and the weakening of the focusing muscles becoming weak. Okay, so this is again, so you have to keep the holding objects further and further. Okay, so these are the classic abnormalities that can now be easily corrected. Now, with all due respects to Ben Franklin, some of the earliest discovery of using lenses as reading eye was, by Jesuit priest, that goes back to as early as the 14th century depiction of a reading glass. Okay, so this were Nicholas de Proville. Okay, and he was at the Great Sorbonne. Okay, and so he had used, so these glasses have been discovered, in the antique. So, but again, Franklin gets the appropriate nod for it. And these are all, I'm sure all of you have these color vision charts. You can see that color blindness is unfortunately very common in males because it's, it's sex-linked. And this was discovered by the great English physicist Darwin, and who himself was colorblind. And he was a polymath par excellence, and he was a proponent of atomic theory and a very modest person. And this may shock you, as his funeral in Manchester was attended by as many as 400,000 people. Okay, and again, colorblindness was very important with the advent of railroad because it was responsible for train collisions, and and there is a small island of in the Pacific called Tinglapp Island where almost everyone is colorblind, so as to how it's genetically transmitted, that thing. So that can be a real problem for, so. And now a word or two about cataract surgery. Cataract surgery has been around for centuries, proposed by the great Galen. Okay, and again, more systematically described in the Ayurvedic literature and called Sushrita Samhita, and in the 5th and 6th century, and then the technique was transmitted by the Arabs to the West, and then the initial one was just a somewhat of a crude procedure of pushing the lens away, and that created that cured the blindness per se, but again was left with a huge deficit that you required thick lenses. And then later on in the 19th century, lens insertion came into out, and now there are varieties of technologies that are available, so-called phacoemulsion with ultrasonic laser eyes. So again, so this is, so the cataract surgery is a passage of light. This is, and interestingly, the leading cause of blindness is not cataract, because it's United States, obviously, with the wide availability of surgery, cataract is number 2 for cause of blindness. Almost at any age, they can be done with relative quite safety. The age-related macular degeneration is a more common cause. So is diabetes and glaucoma. While on the other hand, in the poorer countries, cataract is still the cause, and sometimes people cannot afford glasses, cornea, and the varieties of infections. And there was a type of parasitic infection called trachoma, which was very common, especially in sandy areas, dry eyes. So that was a common cause of blindness, which caused corneal opacity. Okay. And now a word or two about macular degeneration. That's important as to how you can lose the central vision, as you can see from this image. And then the ball and the orange look blurred. Okay, and there are loss of central vision, very common. So there are so-called dry and wet macular degeneration. That is common treatment is variety of anti-VEGF therapy and vascular endothelial growth factor that injected directly into the eye to prevent the growth of these abnormal blood vessels. And this have to be repeated every 3 to 4 months, but they work quite well, I think. So there are varieties of drugs that the 2 drugs that you may have heard about is called Lucentis and Beovu. So these are the so-called monoclonal antibodies. And there are varieties of other photodynamic therapy that is being used as well. So varieties of complex treatment that are available. And there has been question about how the price has been, where people can use, a physician can reduce the cost by selecting the right drug. So that is a big business for treating macular degeneration. And recently, there has been enthusiasm for using gene therapy for macular degeneration. This is the so-called in vivo gene therapy, that the gene is tagged to and attached with the virus, is injected. I'm sorry, I don't have a magnifying lens on this one. Okay, but I can just see if I can get annotation here. Okay, so this is the lens and how healthy eye. This is the macular degeneration. The immune system starts to attack the photoreceptor cells, and when the gene therapy is at the back of the eye is injected with generally an adenovirus with a synthetic gene, and that makes the new protein formation and aimed at stopping the immune system from attacking the cell. So this is how the gene therapy has been tried for the AMD. Okay, and in African Americans, glaucoma and diabetes are much more common, by the way. Okay, and okay, and now this is one way of trying to diagnose. So this is technology which is called optical coherence tomography. It's a non-invasive way of eyes do not have to be dilated, and that can diagnose varieties of abnormalities of the eye called macular cyst, retinal fluid, eye detachment. They can all be so let's just show you this for the sake of interest.
Inside the machine. The OCT can image the deeper layers of the retina beneath the surface, where the early stages of macular degeneration and disease start. The earlier the disease is detected, the sooner it can be treated, and the better the visual outcome. This OCT scan of a normal retina clearly shows all the layers to be healthy. In this image, the jelly, or vitreous, in front of the retina is pulling away from it, apart from at the central macular region. This is called vitreomacular adhesion. In these next 2 OCT scans, the vitreous has moved forward further still, taking part of the retina with it. This is called a macular hole and results in very poor vision. It is possible to treat vitreomacular traction surgically to prevent macular hole formation. This 3D scan shows a blister of intraretinal fluid that has occurred following cataract surgery. The location of the fluid is shown clearly on the 2D OCT section. We treated this condition with steroid eye drops until a normal retinal profile returned. This patient, shown in the next two images, has developed an epiretinal membrane on the retinal surface. As the membrane contracts, it causes distortion and cysts of fluid to form. This requires urgent surgical intervention. In wet macular degeneration, unwanted blood vessels grow into the retina from the deeper choroidal layers. These unwanted blood vessels can hemorrhage and leak fluid into the retina. This can result in massive visual loss if left untreated. The watery fluid inside the eye drains away at a point where the front of the eye or cornea meets the iris or coloured part. If the gap between these two structures gets too narrow, acute glaucoma results. If spotted early, simple laser treatment can prevent the disease. In glaucoma, the transparent nerve fibres that take the visual signal to the brain atrophy away. The OCT scan can visualize the thickness of this transparent nerve fibre layer, and so atrophy can be detected early. Glaucoma caught in the early stages can usually be treated by taking a suitable eye drop to lower the pressure in the eye and prevent further damage to the optic nerve. We believe that all patients over 60 years of age and those in high-risk groups should have an OCT scan regularly, at least every 2 years. Thank you for watching.
Okay, so that gives you an idea about the variety of disease processes. And we talked about the drug for anti-VEGF therapy. So the controversy was the cost of the more commonly available cheaper drug called Avastin and the more expensive unit dose called Lucentis. So there was a huge difference between a few $100s to thousands of dollars type thing. So this is again, the typical problem that we have with American medical system. And again, so these are the classical, the three colors that most of us are fortunate enough to have: the green, red, and blue. And the rods are the black ones, and the blues have the frequencies. You can see that the shorter frequency, the reds have the larger frequency. And now there's, and so dichromates, like the dogs have, they're able to see only about like 100,000 colors, while trichromates, we are lucky enough to have 1 million color combination perceptions. And the color vision has evolved twice in the old world monkeys and the new world monkeys, because of the availability of fruits. Okay, so this is, the new world monkeys were dichromates, I'm sorry again. So opsin is the pigment, okay, and so, and this, again, the color thing goes back to Thomas Young, the wave physicist who proposed the wave formation of light, and to the great Maxwell, who formed the Maxwell's equation. And as far back as 1851, they took color photographs and varieties of combination. When one is stimulated, the others are inhibited. So these are the varieties of cell that occur in the relay center of the brain called your thalamus. And the red and green are mostly in the fovea, which is the center portion. Okay, and while the blue is outside the fovea. So one can say that evolutionary blue is the older gene, and as different evolutionary morphology of the proteins that took place would have the more red and green color vision. So the numbers are different, okay? So that's depending upon the evolution, as human beings have, we have a better night vision that has evolved, okay? And you can, it is so good that people can perceive a small candle as long as 10 miles away. Now, again, your eyes can fool you. So this is like the so-called hue of the color, okay? And this is Maxwell was the first one to take this color photographs, okay? And this is the simultaneous contrast. The top one is the so-called hue of the scent. And you can just see that they look different, okay? But they're exactly the same color, okay? And similarly on brightness, if we cover them up, unfortunately, I think I can't show this here very easily, without messing up the slide, that they take my word for it, that both the center part are exactly same. Similarly, in saturation-wise, this is not less green, the center portion, they're same colors. The bottom one is the saturation, okay? The middle square on the right looks less red. then the middle square on the left, okay, despite that they're, exactly same type of thing. The center is about the brightness. Again, the middle square looks lighter, okay, and so they're, they're again very bright, the same way for saturation, okay, that looks more saturated than less, but again, your eyes can fool you, so to speak, okay. In the same way, now this is a word or two about the so-called afterimage. After looking at the bright object, if the eyes is fixed on the white surface, then the afterimage appears in the complementary color. It's called negative afterimage. So look at the dot. Here's a little tiny dot on the nose, for about 15 seconds. Okay, don't know if the slides will do the justice to it. Yes, I can see it. Now look at the image. You can see the afterimage appears on the visual illusion that takes place in the light and dark area of the image after looking at one image for a prolonged time. Okay, and now these are the varieties of slides of bionic eye that I think I may have that as a thing. Initially, I thought I may not be able to show the video, but okay. Now, again, certain animals have a different ultraviolet makeup in a bright light, and that has been used very usefully in paint on the car and identity theft protection, and so this has been used commercially, interference of the colors, by weakening of some other wavelengths. Okay, hopefully this will show and will not create a strike for.
Medicine man who's been blind since birth shows us you don't really need your eyes to see.
We're going to make a left-hand turn into the grass.
Kevin Jones lost his sight as an infant after being born 3 months premature.
These are the glasses with the camera in them.
Now he's learning to see in a different way.
It helps me understand things that were not possible before at all.
Kevin's been using the BrainPort device for a year now and experiencing some pretty amazing things.
The thing that was on a whole different level for me was watching a lunar eclipse. It just started getting smaller and smaller. It was so cool.
And he saw all of that through his tongue.
The brain is not hardwired, so you don't have to go, for instance, from the eye through the optic nerve to the brain.
Bob Beckman is the CEO of WeCap, the Middleton, Wisconsin company that developed the BrainPort technology.
The BrainPort is more like learning a new language than it is like learning to see.
We had Kevin walk down the sidewalk to show us how it works. The device in his mouth is connected to the video camera on his glasses. Here's what the camera sees. That visual data is sent to Kevin's tongue, which then delivers it to his brain. Now look at the image on the right. That's what Kevin sees.
It takes training, it takes time, and even Kevin is still learning to use the device.
BrainPort draws a picture on Kevin's tongue, and he has to figure out what that is. And once you do, BrainPort opens up a whole new world. In this video produced by WeCab, Kevin is throwing darts at a target, putting a golf ball. He's even able to stop a rolling ball. But the best part for Kevin, using this tool to get through daily life.
You can see sidewalks, you can see crosswalk lines, which is great for really busy or wide streets.
And what's been fun for Kevin, building a visual understanding of the world. He's figured out how to pick up a soccer ball.
I used to miss all the time when I try to just see the ball and then aim for it.
So he started using his hand as a marker.
I was imposing my hand over the top of the ball.
Were you able to see your hand then?
Yes.
The BrainPort?
Yeah, and then when it lined up, then I knew I could reach for it.
And then there are the things explained to him.
Okay, so that was interesting. Okay, and this is another thing called night vision. This was again interesting. Hopefully that will play.
A group of researchers has given mice the ability to see in the dark. Let's see how they did it. Mammals, including mice and humans, can only perceive light waves between 400 and 700 nanometers in length. The researchers' goal? Give mice the ability to see wavelengths outside this spectrum. They started with so-called upconversion nanoparticles. When excited with near-infrared light, these particles emit green visible light. Why not try attaching these nanoparticles directly to the photoreceptor cells in the mouse's retina? To get things to stick to each other, you generally need glue. In this case, the glue was a protein called concanavalin A, known to bind to photoreceptor cells. The inventors added this protein glue to the night vision particles to make them sticky, then injected them into the retinas of a whole bunch of mice. And they stuck. The only remaining question was, did they work, and to what extent? On the left, we have our control group of normal mice, and on the right, we have our group of mice injected with the photoreceptor binding upconversion nanoparticles. First, near-infrared light was shined into the eyes of both groups. The control mice's pupils showed no response, but the injected mice's pupils involuntarily constricted. Next, both groups of mice were placed in dark boxes, where they were conditioned to associate visible green light with their feet being shocked, which led them to freeze in place. After the training was complete, both groups would freeze in the presence of green light, even without the shock. But when the room was illuminated with infrared light, only the injected mice froze. They were clearly reacting to something that normal mice could not see. Next, the mice were placed in a water maze and trained to associate triangles of green visible light with platforms they could stand on. When the triangle and circle were illuminated by infrared light, the normal mice chose their path at random, but the injected mice reliably chose the path with the triangle at the end. Their infrared vision capabilities clearly allowed them to distinguish between shapes. Another version of the task set the green shapes against an infrared background, and the infrared shapes against a green background. These tricks didn't confuse the injected mice, though, indicating that they could discriminate between the green light present in the environment and the green light emitted by the particles in their
Okay, so you can see how there's varieties of technologies that are being experimented. Okay, and this is called synthetic iris, which is again, a dramatic discovery. Sorry, I have to adjust the. Right, it's escalating. Sorry. Okay, so this is not that, I'm sorry that it's not that impressive, but we'll just move on to the next one. Okay, and then now this is again seeing around the corner. Now, this is mind-blowing as to how they.
That will set a few light signals going in lasers, in light-emitting diodes, and those lasers will communicate from one place to another place via a light path, a light guide. So let's press that button. Watch me. We're going to press the button up here, and the laser will fire, and there is the light going around corners. And it's traveling around corners via this effect called interference. It's a lot more complicated a system. Now it's like a two-dimensional or a three-dimensional type of system. But light can go around corners and up or down or across or backwards or forwards, any way you want it, if you can control and if you can manipulate light. Now, it's not just the natural world like beetles and animals that are teaching us how to do that. Scientists, male professors, female professors, all these technicians, all these postdocs and postgrads, men and women. Don't forget, loads and loads of women are in this photonics industry. Previously, physics was limited to male and to sort of stuffy old men. But now, some dynamic and bright young women are enthused and are pursuing these goals. It's a noble goal. Applying the ideas of natural systems to the physical, to the real world, to the industrial or technological world.
Okay, so you get the idea. So that's another way of technology. Okay, and now these are some of the illusions that we talked about, type things. So these are some of the paintings that are in the classic. But for sake of time, I don't think we'll dwell too much on it. This is the great Oedipus, talking about the blindness. Okay. And we'll talk about bioelectricity in some other classes, type things. And these are some of the optical illusions, which are interesting, but not that critical, so to speak. What you think, what you see is not you see, type thing. So again, like how both the circles are exactly the same size, but your eye fools you. The same way, the perspective of the image and the artists have used this to very strong advantage. These are some of the geometric illusions, that they and the line A and B, they appear, they're of the same, appears that they're same size, but actually the A is shorter, okay, and inverted T-line illusion. So even when bisection, the vertical line is still longer, so to speak. So this is how these are the varieties of geometric illusions that take place. How the horizontal line fools our brain. Okay, the same way the Ponzo illusions, which are the identical line within a parallel of converging lines. Okay, as you can see how they look like, oh, they should be different. The bottom one is shorter, but no, they're the same size. Okay. And so we interpret the converging side as parallel lines receding into distance so object to get further away, so to be larger than the nearby object. So the context is important in terms of our perception. So this is, so-called classic Ponzo illusion, okay. And this is also, you can see how that just fools you type of thing, but no, they're the same size. Similarly, there are another classic illusion called Poggendorff illusion. Okay, as the acute angle is reduced, the illusion sort of weakens. So even with, so okay, I think I don't have too many details of this, but I think just for sake of time, I'm going to eliminate this. Okay. And these are some of the better illusions. The rotation of the wheels when relative to the eye movement. On steady fixation, the object vanishes, otherwise it feels like it's rotating basically to us. And so these are some of the illusions which are more recent creations. Okay, and I think we're coming to the end, but I will just show you the top 10 optical illusions.
Creepy. This is a famous drawing created in 1892 by Charles Allen Gilbert called All is Vanity. And depending on what you see first, it can come off as a little creepy. Most people who look at it don't even realize that the drawing is much more than it seems. Have you noticed anything unusual about this picture? You have 10 seconds to figure it out.
Oh yeah. The two women.
Those who look at the drawing notice either a woman sitting in front of the vanity mirror or a spooky-looking skull. If you've managed to spot both of them right away, congratulations! Your visual perception is pretty impressive. Alright, here are hidden faces. Here's another challenging image where you have to find something that's hidden right in plain sight. Now at first glance, it might look way too easy. After all, everybody can clearly see at least 4 faces in the tree branches. But like any optical illusion, there's a catch. See how many more faces you can spot in the next 10 seconds. If you found 10 faces hidden among the branches, then you're an extremely observant individual. Some say that these faces are actually those of famous public figures. Can you recognize anybody? Share your ideas in the comments below. A fair lady. This gorgeous lady must be a ballerina. Otherwise, how does she keep rotating like that with that much grace? Like on her toes? But we're not here to discuss her artistic skills. Instead, can you tell if she is moving in one or two directions? So, what do you think? Left, right, both? The correct answer is she is moving in the right direction. Cafe wall illusion. The name of this illusion was inspired by a tiled wall of a cafe in Bristol. Like Starbucks? Nah. But I will tell you no more for now. Can you tell if the dark lines are parallel to each other or inclined? You have 10 seconds to figure it out. Again, it seemed like an easy question. The lines are so obviously inclined. It turns out that they're not. Let us blur the image quite a bit. More. Now that all the small elements are completely blurred, you can see the dark blue lines are actually parallel to each other. Well, that's not fair. This phenomenon is known as the cafe wall illusion. It was first described more than 100 years ago, but got its name only in the 1970s, when a guy named Steve Simpson noticed a similar effect on the wall of a cafe in Bristol. Little black and white bricks at the intersection of wide lines make this illusion possible. Curved elements inside the blue lines only make this effect stronger. The reason we see this distortion is that our retinal neurons and neurons in the visual cortex work together in quite a peculiar way. Number 5. The impossible triangle. Meet the impossible triangle. But wait, what makes it so impossible, you say? You have 10 seconds to figure it out before you hear the explanation. The impossible triangle, known as Penrose triangle, is so called because it can never exist in reality. If it did exist, it would be absolutely contrary to the rules of Euclidean geometry. What is so wrong with this figure? Well, if you follow the ball sliding on the surface of the triangle from the top point, you will see that the left side is extending away and the right side is leaning towards you. However, they both seem to be on the same plane. Only an optical illusion makes it seemingly possible to the human eye. Now, number 6. Bicycle. Okay, this one seems pretty plain. There is a bicycle and it's moving. Still, there is something wrong here, something mysterious. Can you figure out what the trick is? You have 10 seconds to do so. What makes the bicycle move? Obviously, the fact that the spokes in its wheels are rotating. Nah, it can't be that easy when it comes to optical illusions. In fact, the spokes remain in place, and the colors that fill the wheels, those 50 shades of gray, that keeps changing. This illusory motion effect is created thanks to small, quick eye movements. Scientists are still working on figuring out how this phenomenon works to understand the perception of motion better. Now I present to you Rubik's Cube. What are these red and green and white lines all about? It looks like something familiar, but what exactly is it? If you need more time, just pause the video. We are going to need a cylindrical mirror to get the right answer. It's a Rubik's Cube. This optical illusion is based on anamorphosis. Hey, I remember anamorphosis. She used to live down the street. Actually, it's a form of perspective. The idea of it is to transform images so distorted it is almost impossible to guess what.
Sorry, I think I...
The idea of it is to transform images so distorted it is almost impossible to guess what they are into something that makes sense using a cylindrical mirror. And it only makes sense if you do it from one special point. so only one person can see it at a time. It would be a great way to send hidden messages. It turns out this technique is not a 21st century 3D era invention. It actually dates back to late Renaissance, which was in the 16th century. Chessboard. You can't play any chess with this board because it's moving all over the place. Or is it? You have 10 seconds to do so, and please try not to get hypnotized. As hard as it is to believe, the eyes actually remain where they were. You can see it if you focus on just one of them. If you stare at one point and ignore the constant movement of black and white dots, it all becomes clear. And that's the way, huh? Counting down from number 9, Upside Down Steve Buscemi. Can you tell me what's wrong with Steve Buscemi? I mean, I hope Steve is perfectly fine, but can you tell me what's wrong with the second picture? These pictures look the same to me. Hit thumbs up if you think so too. Let's try to turn it upside down. Oh no, that is way too scary. It seems like my eyes tricked me again. By the way, here is another fun celebrity optical illusion. Hermann grid illusion and scintillating grid illusion. Look at this black square crisscrossed with perpendicular white lines. The white dots situated at the intersections of the grid shift their color from white to gray and back. When you concentrate on a particular dot, you see that it's white. But as soon as your attention wanders, the dot turns gray. That's the Hermann grid illusion. Amazing, isn't it? We can take this illusion one step further by positioning white dots at the intersections of gray lines, all placed on top of the black background. Look at this image attentively, and you'll notice black dots starting to pop up at the intersections of the grid. Doesn't it create a scintillating effect? Another name for this illusion is a simultaneous light contrast illusion, as you perceive the dots as white at one moment, and then, almost immediately, you see them as black. How is it going? Have you managed to spot this effect?
Okay, so I think we've seen this, and that was probably the end of it. So I think hopefully you had some fun learning about eyes and vision. And I think next time we'll continue on senses and hearing, and followed by wrapping it up with smell and taste. Okay, folks, thank you. It was fun recording this. Bye-bye.